Thanks, Tanya. Thanks to all the organizers. It's been wonderful. So today I'll tell you a bit about the games we've been playing on cell division and filaments that need to die for the cell to divide, which seems to be universal. And I'll start, I guess, with background, which is cell division is the foundation of life. Basically, we're all a colony of one cell that divided a long time ago and kept on dividing. And it is inherent in the physical process, right? You need to take a droplet and make two droplets. And across evolution, that's done by protein filaments, which assemble and disassemble, as I'll show you, and are coupled to a compartment and, of course, an energy input because they're assembling and disassembling. And now you might think there is like a million ways by which we might split a droplet into two. There's actually only three main ways. And they're really split by the tree of life. So this is the tree of life. It has two prokaryotic branches. So these are organisms that don't have internal structures. We call archaea and bacteria. And then eukaryotes, which is us, emerged as a symbiosis between these two. And I'll speak about this in a couple of months in the few slides. So imagine if you have like a compartment to divide it. First, you need to grow it. So I will not speak about that. And then there are two ways by which you can divide it. You can either pinch it in the middle, which is what archaea does. So archaea uses, at least those archaea that are closest to us, they use something called the escort filament, which are long and flexible. They have active geometry changes and they somehow squish the cell into two. Another way to split a compartment into two is by building a wall in the middle, right? By building a partition. That's what bacteria do. So what they do is, again, use filaments called FTZ filaments that, unlike these that are long and flexible, are short and stiff. They treadmill, meaning they, as we've learned from Karsten, grow and shrink with an equal rate. So it looks like they're running around, but they're really not. They're growing and shrinking. And this treadmilling is somehow coupled to the synthesis of the material in the middle that's going to split the cell into. Okay, and then today I'm going to speak about both. This is a bit older, but in my mind, it was part of the story, so I had to mention it. Then I'll speak a bit more about this part of active matter. And then very briefly, I'll speak about this merger between archaea and bacteria. So now that, after decades of phylogenetic work and on, we know that most likely eukaryotes emerged by archaeal host. So host cell here in blue, growing around other archaea and other bacteria. Bacteria became our mitochondria. So our mitochondria, has its own division, has its own genetic material that we got from our moms, and it's kind of a separate organism within us. While archaeal host gave us our nuclear membrane and our outer membrane. And I really like this picture from a recent review, where you have an archaeal cell that grew around other archaea and bacteria. So this will become our mitochondria. And now here you see there is the nucleus becoming with double membrane. This would become our ER, which is continuous with the nucleus. And these guys will fuse and become our outer membrane. And we know actually that our membrane reshaping machinery in eukaryotes completely comes from archaea. So everything that's set in archaeal membrane is now in our nuclear membrane and our plasma membrane. And what kind of just to for completeness, how eukaryotes now divide, given that we came from archaea, we still use archaeal machinery, these are these escort filaments, to cut when the eukaryotic cell is squished to about 1 micron, which is the size of archaea. But then, because the eukaryotic cells are much bigger, they had to invent an additional mechanism, which is this optimizing mechanism, which acts at a larger scale, around tens of microns. So optimizing, which we also started to work on very recently, and Karsten nicely explained to us a lot about it, somehow squishes the cell, but only up till around the micron, which is an escort that we got from archaea kissing. Okay, and kind of in the recent years, this field of evolutionary cell biology has bloomed. And in particular, people have managed to culture symbionts between archaea and bacteria, and special archaea that are closest to us genetically. And the way they look like, these are isolated from the bottom of the ocean in Japan. These are actually from Slovenia, but it took them this 12 years, this around 5 years to manage to grow them and then image them. And the way it looks, it's quite remarkable. there is an archaeal host that then grows tentacles around bacteria and other archaea. And as I told you, this is to become our nucleus, this is to become our ER, and somehow, after 2 billion years, they'll give rise to eukaryotes. And in a very recent Nature paper, there were beautiful cryo images of this symbiosis and also actin that's inside, which, however, I should tell you, there's no motors in archaea, so it's probably not traditional activated. We don't know whether it's treadmills, but I'll briefly speak about this in the last couple of slides.
Sorry, but actin in archaea?
Homolog of actin in here. There
Not in bacteria.
it also is used in bacteria as MRE. I mean, there's been horizontal.
Sorry.
The current hypothesis, just to comment on that, is that the last common ancestor had everything: actin, escort, tubulin, and then it's kind of specialized.
Back to the vision. So there, the textbook image, of course, is the actinomyces and forms can knock out already, but that's being questioned because there are experiments that show you can knock out the myosin.
Exactly.
Okay, so
Yeah, we know about that. We that's what we are currently working on. We don't know exactly. I mean, we have some hypotheses, but it's very preliminary. But definitely the ring forms without myosin, because in our favorite active matter picture, there's this picture that filaments flow, and then when they collide in the middle, they would align. But now we know that the ring forms without myosin. So you don't need that kind of activity. You do need ATP for turnover. You do need gradient of rho A. So you need something that's going to position in the middle and also cause it to grow faster and nucleate faster in the middle of the cell. And I think that's probably the key. But yeah, you don't need motors.
Also for bacteria, you're focusing on gram positive, so gram negative.
Well, both FTZ is in both, but I will not speak about wall building. Still, I'll just speak about the ring formation.
Okay, just continue this. I mean, so I can see the ring formation is an important part. What do we know about the process of all the way to the final constriction?
Yeah, well, very little actually. We just got 8 years long grant just for that. But we know there's, we know the treadmilling and this ring formation is needed for the division. Is needed to place the synthetic machinery at the right place for distributed. But then once the division has started, even a little ingression started, you can knock out the treadmilling and it still progresses. There are a bit funnier shapes, but it still divides. So it's not necessary for division. But we know that you need to deposit material somehow in concentric circles from outside towards the inside. And probably the last pinching is done by another machine that not known.
Yeah, it's hard for me to think that there can't be anything else.
There's probably another machine that pinches the membrane at the end. Okay, so it's mentioned today. I'll tell you briefly about the division of archaea, that is flexible, elastic, active rings. Then I'll tell you a bit about the formation of bacterial division ring. And then in the last couple of slides, I'll tell you about our very recent gains on obtaining morphologies of different symbiotic organisms. Okay, and first to mention all the heroes of the work. So these are 2 generations of PhD students and postdocs in my group who did everything. This is archaeal and escort team, bacteria, and symbiotic morphology postdoc, and even a little bit jumping in areas. And all the biology, all the experiments have been done by 4 groups: Buzzbaum at LMB Cambridge, who taught us everything we know about evolution, and also archaea and escort. Also escort in eukaryotes has been done by Aurelian, which I'll not speak about in details today. And bacteria in vivo and in vitro has been done by Seamus and Martin's lab. Okay, so let me start with archaea. So what Buzz here in Cambridge managed to do is they managed to live image the closest archaeal relative that's easy to culture. So these are not these tentacle organisms, but they're quite symmetric organisms. They live at extreme conditions. This particular one is called Sophilobus and comes from Yellowstone National Park. So it lives at 80 Celsius pH 2. And it took them like 2 years to build a microscope to do this live imaging. So I'll show you what I found. This is the cell. The DNA has somehow been divided, we don't know how. And then the way these cells divide, they build this escort filament. So, escort is just protein. And they build composite filaments. So first, they build one. There's one escort type, which is in pink. Then they build another ring on top of it, which is just a different isoform of the escort 3 protein. And then this stays together, and nothing really happens until they consume energy for disassembly. So they chew up by an ATPase, this bottom filament. And then once this bottom filament, the template has been gone, the green one that is on top somehow squishes the cell into. So, we being top metaphysicists, we really like this picture and thought about maybe 2 elastic filaments that work together. So the first filament would be something stiff in our mind that matches the cell curvature. But then the second filament would have a lower radius curvature, but can bind only when the first one is present. So it's going to be stretched like a loaded spring. Then together they are put. And then once the first one is gone, like remote stopcock, the second one is released. You release the spring and it squishes the cell into. So it's 2 filaments, different curvatures.
Can I think of it like a rubber band?
It's something exactly. Yeah. Oh, in our soft matter minds. So to test whether this is physically possible, we build this model. So we have a cell, which for us is just a membrane vesicle. It's built out of this 1 bit thick membrane model, which I can discuss in more details. Which actually I will not discuss much of simulation at all, like the ingredients, but I'll happy to speak the details at any point. And then we build a filament in the middle that matches the cell curvature. So we build it out of these 3 beaded units, which we realized the minimal ingredient to have chiral filaments, and they're connected by springs. And basically, these the springs control the curvature. So you see, there's nothing optimistic here, it's just a filament. In doubt, everything is under Jones interactions, and we run larger than it.
So what about the attachment of the filament to the membrane?
Exactly. So these blue beads attached to the membrane via, again, for us, Lennard-Jones, attractive interactions. In the cell, it's an electrostatic absorption. It's really driven by surface absorption.
So it's really electrostatic.
Right.
So, and you show that by changing salt concentration to show that how do you know that it's electrostatic?
Well, then we know that not necessarily for archaea, but we know for human, as what people have done in vitro reconstitution.
Okay, so
there are also genetic simulations there. For human escort, it's been done quite well.
Okay.
So Andela, you are looking at the case where this filament has formed and it's kind of pinching, but are you also looking at the formation of the filaments, or that's kind of say?
Exactly.
Besides modeling the beads, do you make an effort to conserve the volume of the vessel?
Yeah, so not in what I'll show you, but we were worried that caused an effect. So at some point, and it's in the side of the paper, we filled it up with beads to conserve the volume. And then it didn't change the mechanism, it just changed, you needed more you need a stiffer filament to squish it because you're squishing at this higher pressure.
I have another algorithm we can talk about.
Yeah, for sure. Okay. That'll be great. Yeah. Right. This was not very quick. And also you can, it's just sorry, you can fill it up to like 25% because then they start crystallizing with the squish and so on. So you cannot control it super precisely.
Okay, I'd love to talk to you about it.
And the filament don't open the surface.
Not so much. I mean, in principle, yes, but it's bind quite strongly. So we don't see it diffusing much. And we don't see it also diffusing in the cell, which I'll show you. Okay, and then to model this idea that when you remove the first filament, the second one reaches its target curvature, we suddenly change the curvature by shrinking these bonds. It's the step we put in, and we expect the filament to go from a ring into that helix. That's how we compute the length of these bonds from side by side, and then
so that shrinkage then is like a power stroke in a motor.
Exactly.
Right.
And
presumably ATP or something.
Yeah, we know that there's an ATPase called UTS4 that chews up this filament. I'll show you pictures when we knock out this one, and then it doesn't disassemble. But how you do it, as you may feel, really matters. How you put this power stroke. Stroke will control whether you divide, how symmetrically, how efficiently, and all these things. Okay, so I'll show you what happens. We start with a vesicle, filament is happy there at the equilibrated curvature, and then we suddenly change the curvature everywhere. And what you see is happening, the filament coils up, squishes the cell, but of course it stays there. So you need to also disassemble it to be able to divide. And kind of disassembling the cell is equally important to assembling. And we know in the cell, actually, if you measure the content of this protein in the cytoplasm, when the cell is constricting, it goes up, it's disassembling. And then we also have more recent knockouts of these ATPase that disassemble the filament. And then you very nicely see the filament starts growing very long, and it's kind of phase separates the two filaments according to their curvature. See, the blue one stays preferentially in the middle and has a smaller radius of curvature, and the big one, previous green, kind of goes on the outside.
And this is simulations.
No, this is experiment. This is super resolution imaging.
I'm sorry, in the simulations, the filaments that the outside are they always the inside? Inside. Okay. So you've got basically very thin membrane around.
Yeah, exactly. It's just visualization, but they're always inside. So what we did is start the disassembling by chopping out the bonds. And of course, now again, it matters how you do it in the non-equilibrium case. If you do it too quickly before the membrane is managed to push, then it will be as if nothing happened. But if you do it with the right rate, so the filament spoiling, it's squishing, and then you slowly disassemble, then you get the tight neck. And in our case, the membrane naturally cuts, just like thermal fluctuations. We didn't do anything to cut it. We can discuss whether these kind of coarse-grained fluid sheet membranes are good enough to represent scission, but energetically it realizes that this sapphire is too big spheres rather than a very tight neck. Okay, so we're happy about that, but then what I thought was a real value of these simulations is that now we can start comparing to live cell data. So we got a bunch of movies from Buzz on these archaea dividing. And we taught ourselves some image analysis together with our super resolution microscopy colleagues. And what we did is we measured the cell neck as it's squishing. And of course, you quickly learned that cells are living organisms. They can have different sizes. They can take different time to divide. But once we've learned that we can normalize the neck by the initial cell size and time by the division time, we get all the experiments, and this is like 70 cells, fall onto the same curve, which we as physicists love. So this is basically the neck size scaled by the initial cell size, and time is scaled by the total division time. So all the experimental data falls onto the same curve. All the simulation data falls onto the same curve, but they are not the same curves. So clearly, in experiments, you start changing your filament, but the membrane lags, so nothing happens, and then it's on a linear decrease, and then it tails off. While in simulation, as I told you, we change the curvature everywhere. At the same time, you give it a power stroke, it spread real, and then you squish very quickly. So probably we should start thinking about the rates of how we change curvature. And also, you might have noticed that our filament, I told you, what we put in is that it should go from ring into tight helix. But what actually happens, it creates all these reversions, the inversions of chirality. It's called the hemihelices. It's basically supercoiling, and it's a bucking instability that people see across the scales. It's from a hydrogen black on the shape of the guts, which is a bucking instability of two elastic materials that have differential growth. We had huge number of discussions with our colleagues from the LME in Cambridge saying that this might exist at the macro scale, but not at the nano scale. And then we actually went back to cryo data from human escorts. So we don't have cryo structures from archaeal escorts yet, but we have human ones and yeast. And actually what people report as helices are helices with a bunch of these inversions of chirality. And we think they might be functional to shorten your filament, to basically drive constriction. And it's the same physics as coiling of your Christmas ribbon. And here you might also notice, if you coil your Christmas ribbon quickly, you'll get a lot of these perversions. And that's kind of our case. We shrink everywhere at the same time, so we do it infinitely fast. But if you curl your Christmas ribbon slower, you'll make a nice helix. So we should probably, once we think about the rates, if you do it slowly, our filament will have different shape, and kind of the membrane response will be different.
So your model is a model where you basically you go from having one sort of minimum to another, and whilst the real system is probably you have these two things which are probably one of them is under tension, and then the other one disassembled. So in some sense, your initial ring is already has some bits which are under tension, is maybe.
That's probably true, but also these ATPase that choose up, I mean, our coiling is really modeling these ATPase that choose up the templates. And
Yes.
that's probably not infinitely fast. It probably goes with certain rates, and how you do it also matters. Like whether you chew it up for one end or for both ends or randomly. So we tried all the different protocols, and they really do influence the shape of this curve.
But the fact is that there's also the tension, which is, which might also play a role. The fact that you have somehow stress, there's some internal tension in
Which might not also be uniform. You're saying.
Exactly, yes.
Exactly. There is a hypothesis that these ATPase attack spots that are more on the tension because proteins would be more exposed there.
yes.
And this ATPase is quite violent. It unfolds the whole protein. So probably those that are not exposed,
Those could be
hexagons.
exactly.
Exactly.
So if I think of your whole simulation and the experiment, so should I think of the experimental data as something like it's going to be for minus Q squared or something, and that one just having for minus Q.
Yeah, I mean, we tried fitting it, but you can. But the thing is here, you come to very tight nets and your measurements become very noisy. You start hitting the resolution limits. So I wouldn't. Yeah.
What would we make of these little wiggles? Is there, I mean, there's of course
Here?
a spread of, but there's spread along the data, but those wiggles are still consistent and those fine structures are there. So.
Yeah, these there is a lot of post processing here. So I wouldn't look too much into it. Maybe there's something that we didn't think, but knowing how dirty these measurements and systems are, I was still quite surprised that they all fall kind of on the curve. And basically just to wrap up, we managed to get the experimental curve only when the filament coils at random positions. It gives the uniform spread of tension and gives the most symmetric and of the most reliable division. And kind of makes sense which ones spread tension uniformly, but it also kind of points out that this ATPase is not working processively from one end, which some groups have proposed, but it's probably jumping randomly along the filament, so that's something that can be tested. Also, if you manage to poison this ATPase a bit, but not completely, which I showed you earlier, you should be able to change the slope of this curve. So these are predictions that we can make. And this is very recent from another super resolution study where you see the membrane in red and this constricting filament in green, which is kind of bumping out of plane. So that could be the supercoil, perhaps. Okay, and just to comment on, so yes, this escort filaments divide archaea, but as I said, us on eukaryotes, we have inherited them. I showed you in archaea, they have 2 filaments. In our cell, there is 11. They can all copolymerize with different stoichiometries. They can have not only ring shapes, but helices, caps, all sorts of shapes. And they do many other things. So they still cut the bridge between 2 cells in cell division, which is around 1 micron. But they also do trafficking. They do binding of viruses, like HIV uses escort to bind. So we worked on that this eukaryotic side, but I'll not speak about that today. I'll put them here as references. And I'll just comment on how we believe that's going to be a new type of force production at a nanoscale. So unlike actin and microtubules that consume energy at the level of individual monomer, here you build a whole dead nanoscale structure, and then you completely remodel it to produce work. And I like this idea about studying how that our machinery works, but going back in time, where there's less components. As I said, we have 11, archaea has only 2, 3, but kind of the physics is likely conserved. Okay, now I'll take you to the other side of the tree of life, which is bacteria. As I said, they use something that might look similar from, if you look from far away, it's a ring in the middle, but it works very differently. So those, as I said, short and stiff filaments that treadmill, so grow and shrink. These are cryo images of the filament inside vesicle. And I'll show you what it looks like in vivo. So this is Bacillus subtilis, bacterium. The FTZ protein will be labeled fluorescently. And what you see is that it's kind of fuzzy everywhere in the cell, then it condenses and the cell divides, condenses and the cell divides. So And this condensation of the ring, so this FTZ condensation in the middle, is necessary for the division. So I'll show you here from rather than this experiment. We have kind of FTZ proteins everywhere along the cell, but at some point they form a ring in the middle. We know that we recruit synthetic machinery for building this wall that's going to split the cell. But we know that the treadmilling, growing and shrinking, is necessary for the initiation of constriction. And today I'll speak about that, just the formation of the ring, not about the whole division, because we really don't know much about that. Okay, so the question is, how does this ring form and why is it important that it treadmills? And now this is much easier than what I showed you previously because we know, sorry, question? Because we know so much about bacteria. You know, they make us sick, unlike archaea. So people have studied them to death, and they also live at room temperature and so on to be easy to study. So for FTZ filaments, we know that they nucleate, they grow on one end with some are on, they shrink on the other end. So what they actually do is they hydrolyze GTP somewhere along the filament. And then once the hydrolyzed monomer related, it reaches the end, it detaches because it has larger mismatch with its neighbor, so it's less bound and it detaches. Okay, so basically the polymerization is governed, or the dynamics governed by three parameters: nucleation rate, growth rate, and shrinkage rate. And you know, if you effectively make growth and shrinkage rate equal, you can get filaments to treadmill. So this is Monte Carlo simulation. They look like they're translating, like your favorite self-propelled filament, but they're actually growing and shrinking. And we know that FTZ filaments in vitro, which I'll show you, and also in vivo, don't diffuse. There is no translation there. There's really growth and shrinkage. Okay, so I mean, this system has been, at least at the level of a single filament, described super well in the literature. So you can test that our simulations match everything they should, theoretically at the level again of a single filament. We can also change our parameters, a few parameters, to match the experimentally measured lengths of filaments and also velocities of treadmilling. So when we match these two parameters, we can measure, for instance, the distribution of velocities and lifetimes, and it really matches simulation experiment quite well. So we are capturing something reasonable. Okay, but it's all on the level of a single filament. Now what we can do is we can study many of them, which you cannot do in the previous kinetic models that are analytical. So what we can do is we start nucleating filaments. They start growing and shrinking. Again, looks like they're translating, but they're not. And what happens very quickly is we see them aligning. Just like, again, you would expect in your favorite self-propelled filaments, but they're not self-propelling. And now the question is, why do they align? And interestingly, at the same time, the Loze group at our institute had this high-speed AFM data, these filaments growing, kind of at low surface coverages and high surface coverages, and you really see that the high surface coverages, they also align. They also measured it in our environmental paper, diffusion, which of individual monomers, it's not diffusing. They also measured correlation between monomers of the filament. They are not correlated, meaning there is no pushing. So FTZ filaments are very stiff. Really the mechanism of their alignment is very different.
Presumably this simulation is 2D.
Yes, yeah, it's all 2D. It's like unwrapping the cell because the cell is quite stiff. So.
Yeah, so you can sort of imagine that steric interactions will tend to align them. The real situation does allow for 3D movement. I imagine it's not strictly bound to a membrane, right? At least by analogy with other cytoskeletal filaments, but
so
can actually cross over.
So again, there are two things there. So first of all, for the steric interactions to align them, they need to diffuse, right? You need to hit and then diffuse. They don't diffuse in AFM, or they're super strongly bound to the membrane by a linker.
No, but as a monomer joins and treadmilling, I realize they're not translating. As a monomer binds, it's still subject to those steric.
Right, but exactly, I mean, steric interactions will be important, as I show you, but it's not as if you were to pack your system with hard rods and then shake it and wait for it to align. because that requires the rods to diffuse around to align. Maybe I'll show you in a second. And then we go back here. And then just to show that we measured the nematic order versus surface density between simulation and FM data, they follow the same curve. Okay, but the question is, yeah, why do they align? And it's really by the virtue of filament dying. And it is volume exclusion that's key here. So what happens is when a filament grows or against another filament under an angle, it will not be able to grow because there's no space and they're stiff. So the filament cannot bend to make space for another monomer to fit in. But it can still shrink, right? So you get the mismatch now. The filaments that are under an angle to their neighbors, they shrink faster than they grow. And what that will lead to eventually is that only those that are aligned survive. And that's a slow process. And I'll show you in vivo also this alignment is slow. This densification of the system is slow because it requires your filament to nucleate, try to grow, shrink, try again, and so on and so on. But eventually it will populate, and only parallel filaments will survive and will run parallel to each other.
So the rates of your polymerization, are they related to any forces on the? No, completely just.
Yeah.
But you could imagine also that actually that they could be related to forces, and then you might even speed this up because then actually if you're going against a barrier, then you're more likely to, if your unbinding rate is enhanced by external stress, that you might.
Yeah, that's a good point. Yeah. We don't have it here. I don't know if it's known.
Yeah.
For
I mean, it should just make this happen faster.
It's definitely this mechanism has been studied for actin.
Yeah.
And then we thought a lot about how different this is to actin. I mean, it's not in the sense that they're both straight-line filaments, but actin is softer. And this whole business that also Rhoda showed of actin growing against membrane and pushing really requires the membrane to fluctuate so that you can fit a monomer, wait for another fluctuation, fit another one, and so on. But you cannot do that here because they're quite stiff. And actually, we do have now recent set of simulations made vary the stiffness, and you can see that if they're softer, they start aligning by pushing. But then you also see them being correlated in diffusing, which you don't see.
If they're softer, then you should not see this kind of nematic order.
No, you do, but the mechanism might be the dominant mechanism, or another mechanism contributing might be this pushing. Because if the filament against which you're growing is fluctuating, it will make a space for another monomer, which has been proposed for actin.
This might be a silly question, but why do they some of them slightly bend along ones?
I mean, they do have some bending rigidity, which which we matched the experimentally measured one. I mean, it's a bit debated also what the experimental measured rigidity is, but we are in the right range, so they do bend a bit, but not enough to accommodate another monomer if you're going against. Sorry, are there any other questions?
Well, that will depend again on the relative angle. I mean, you can imagine coming in at a
for sure, and also
angle and the finite bending together with, there will be some residual fluctuation, small.
Yes,
But some residual, yeah.
exactly.
But also, I'm still a little concerned about the possibility of jumping over.
Yeah, sorry, I didn't go to that.
Which would be like an energy barrier or something that they might occasionally get over.
It might happen, but they are bound. They're membrane bound by a linker, which is finite. Like it's not super extensible. Yeah, they have and actually the linker itself forms another filament underneath, which is quite curved.
But I feel electrostatic.
No, but there still might be some, that locally detaches, goes through and comes back. Like that, it's possible.
And the biological reason for this is probably because the wall is much stiffer. Is that why?
So maybe,
I mean, if I was thinking about why are they so strongly bound to the wall? Is it because the wall is much stiffer? Is that basically?
I think so. I still think exactly, but I still think they do need to cause a little deformation once they to guide the wall synthesis. So you might think that yes, it's stiffer, but they still need to pull it a bit, so they need to be strongly bound. Okay, and then how do we form a ring? Well, you need two more ingredients. So they need to be shrinking. Another ingredient you need for them to align kind of perpendicular to the center, and so this is their curvature. So FTZ filaments themselves are not strongly curved, but when they do make this composite filament with their membrane bound anchor, they are actually quite curved. I think here, radius of curvature is 200 nanometers, so quite curved. So that's what we put into our model is the filament curvature, which will make them go this way rather than perpendicularly. And then, how do they pick the center? So we know the way bacteria measures their size is by spatial chemical gradients. So there's an underlying reaction diffusion going on. In E. coli, that's this MIM system. In Bacillus subtilis, there are other mechanisms, but effectively what they do is they change the local nucleation and polymerization rate. So effectively, when the cell is of the right size to divide, the nucleation and polymerization rate in the middle will be faster than at the poles.
So your model hard codes in the fact that they align along the, but the previous model and this model don't address, if I understand correctly, why they align along the hoop and not along the long axis.
So yeah, previously we didn't have any kind of out of plane curvature. Now we add it.
Okay,
So if they're very curved, they'll just they'll fit more easier in this way than around this way, which has no curvature.
and so the filament itself doesn't have curvature, but when it's bounced to the membrane with the linkers, that's when the curvature.
Yeah, because these linkers themselves polymerize into quite curved filaments.
Okay.
Okay, so now what we do is we have a curvature term, which kind of measures the local bond angle and the local curvature on in the plane, because we represented as basically cell, it's unfolding. And then we also change locally the rates of nucleation growth. We make it faster in the middle of poles. And what this will produce is a ring that runs in the middle of the cell. So again, the mechanism by which they aligned is this growing and shrinking. We do have control experiments actually both in vivo and in simulations. When you stop treadmilling in vivo, there's a drug that can do that. They remain diffused and don't form a dense ring. Okay, so now we put it back on the cylinder and you can see a nice ring forming. It's kind of running around. Just take time. And then what else we can do, which again, I always appreciate, is when we can compare it to lifestyle data. So we got super resolution measurement on the ring width and ring density over time. And you see that the ring width decreases very quickly. And that's really triggered by this onset of chemical gradient. And that's another reason why you might want to be dying and growing, because you can very quickly respond to external gradients. So this gradient comes in quickly in the cell, and that causes the filaments to disassemble at a time scale of hydrolysis and reform elsewhere. So that causes very quick nucleation. So very opposite of traditional equilibrium physics, your nucleation is very fast, but then the growth is very slow because this growth requires you to select against misaligned filaments, and that's a trial and error process that takes time. So you grow over tens of minutes, but nucleates kind of under a minute. And
So your simulations look a bit faster than the experiment.
yeah, especially here, they do have this jump, but we also had a bit of problems, normalizing with their fluorescence. There's choices you have to make there. As I said, it's still dirty, but qualitatively, the picture of fast nucleation slow growth is there. So I think, I wouldn't dare to compare the exact numbers and fine details of the curve. But also, the time scale we get out of it is similar. And yeah, the reason why we, what I think is contribution to biology, can explain why treadmilling is important, because they were, why is it important to formation of the ring, but not for the division? Because this treadmilling allows you to respond to the external gradient quickly, and then it allows you to align filaments that are strongly bound and don't diffuse. And another, I think this is maybe just a fun fact from experiment, what I saw is that the ring, when it's mature, it runs around faster than in the beginning. So they looked into, they had all sorts of hypotheses why with time the velocity of filaments in the ring would be faster. And we naturally observe it also in the simulations, and it's simply because you have less misaligned filaments that have zero velocity. So with time, if only those that are parallel to each other survive, they will all run with their almost maximum speed. So it's kind of just a consequence of the alignment mechanism. So here I've shown you this dynamic growth and shrinkage, which might look like translation, but it's not. Can drive collective filament alignment, and to get a ring, you need to couple it to curvature and chemical gradient. And the ring cannot form without straight milling, because they remain under a certain angle, they don't fuse around, and you get kind of like a jammed, a jammed. And then there is separation scale due to growth formation, fast condensation due to chemical gradient, and flow growth due to the selection against misaligned filament. And kind of more generally from the active matter perspective, we have now a project where we moved this code into molecular dynamics so that we can compare this system of growing and shrinking with the system of self-propelled rods. And they do have different, again, they will produce your aligned filaments. But for instance, if you try to perturb a system of self-propelled rods, that information will go fast, spread far away. You push it on this side, you'll push a filament, push another one. But if you perturb this system, you create a local, defect. These filaments die, and you don't propagate information. So this kind of another. You might want to have both in your cell for different functions. So I think the response to perturbations, the role of defects and so on might be quite different in this system compared to self-propelled filaments. Yeah.
So in the statistics, we sometimes see that there's a helical filament or helical structure emanating from your inclusions in that.
You do get it if the coupling to curvature is not as strong. So that would be if you don't have, if this linkers to the member are not as polymerized. So you can get that. If it doesn't care so much to go perpendicularly, it will also want to go just randomly along, and then you'll get effectively a helix with a large pitch. But that's kind of not a functional structure when it's divides, at least in subtleties, it's very condensed.
So it's when you start to correlate.
Right, yeah, but not for the division. Yeah, for sure. And at that point, when it's doing that, it's not meaning to divide, so it doesn't have a strong gradient that are going to localize it there. And currently, we're still interested how such a ring can cause some small deformation that might kind of break the symmetry and drive the building of the wall inwards. Okay. And then in the last maybe 5 minutes, I'll show you our thoughts on in this process of symbiosis between archaea and bacteria, archaea and archaea. So what we've been wondering is why did all the cells of this closest relative archaeal relative to us that's grown with its bacterial symbiont, why do they have all these long tentacles? And I should say there's, again, no motors that are going to push them. Actin is found inside it, but it's not very dense, and also not all of them have actin. They still grow even without actin. So it could be that actin is there for support or to help diffusion of stuff to the tip, but most likely it's not pushing.
When you say actin, is it monomeric or
Filaments.
It is filament. Okay.
Yeah, I should have put it here. So I might have a cryo structure. So this is what it looks like. This is another tentacle zoomed in, and we see actin here in yellow. We don't know if it treadmills this archaeal actin. Nobody's managed to reconstitute that. This is all from December 22, so it's quite new. And there's definitely a lot of work going on, so I'm sure we'll learn something more soon. So our question was, why and how can this tentacle arise? So there's no motors. It's not fully filled with actin, so actin can push it like in lamellopodia. And also, when sometimes they do have archaea on their own, this they have archaea in the culture, and then it doesn't have tentacles. has tentacles only when it's growing with symbiote. So we started thinking about symbiotic interactions. So I'll show you just two quick slides. So for us, this is a host, let's say archaeal host, and a pink bead, let's say it's a bacteria, and it's stiff and it's a symbiont. So what they can do, they can, interact, let's say attractively. Archaea likes symbiont likes archaea. What you'll get is, then the bacterium diffusing on the surface of the cell, nothing, too surprising there. But what you can also do, given that they have coupled metabolic cycles, their interactions might not be, reciprocal. One might be wanting something from the other one, while the other one might want to escape, like predator prey. Also, we know that lots of these archaeophiles are anaerobic, while bacteria are, in this case, are aerobic. So this guy releases H2, which for instance might be killing it. But so it's the blue one might need something from the pink, but whatever the blue one has might be damaging pink. So the pink one might want to run away. And then in those cases, when you now have asymmetric interactions, for instance, in this case, pink likes blue, but blue doesn't like pink. So what you get is huge tubes, like kind of long tubes on this side. But if you do it the other way around, such that you have a particle on the outside that this one wants to catch. So this one wants to catch the symbiont. The symbiont doesn't really want to be next to it because this one is releasing a chemical that's damaging it. What you'll get effectively is these long tentacles. And then you can, of course, do it at different coverages. So if you do multiple,
number of particles or
yes, and that's another thing. I think to get this long intertwined tentacles, you probably need to grow the membrane area, which makes sense. So we are now adding it to the system. And actually, we found that Suri from Chicago has been also thinking about membrane growth and also some structures that look similar. So we are kind of discussing there. And then, of course, it depends how many symbionts you've got. If you got on a few, you get tentacles. If you have a lot, so you're completely packed with your symbionts, you get something, completely crystal gem. So this is just to say that, under different, but here's things to convey non-intuitive, kind of the value, the range, all of that matters to the shapes you're going to get. So we are now looking through the literature and all different shapes of symbiosis and under which conditions you can recreate those morphology. Okay, so I'll wrap up here. I'll thank my group. Again, this is everyone who's done the work. And Bazer, Leon, Seamus, and Martin taught us everything on the biology side. And yes, we have one postdoc position on bacterial division on the wall building, if you know anyone who wants to play this game with us. Thanks.
Okay, so we've had a lot of discussion already. So, Alison.
Yeah, very messed up. So if I remember in the experiment of FPX there in Europe, I think some images that in the FPX able to form hypothesis instead of just the environment.
Yes, those are mutants that are more curved, those that form. So this is wild type, what I showed you. Those that form the swirls, which was on bioRxiv and is going to be published soon, is mutants that are more curved, and also they're not bound to them. This is filament. This is how it will bind to the membrane. In those experiments, they are bind side on, so they're curved in plane, and they form all sorts of nice, as I said, defects and vortices depending on the curvature. The kind of was to change the curvature of the filament by mutations, and then study kind of their alignment in it. And it's interesting that still the vertices, the curvature of the vertices they get, kind of the filaments that treadmill in plane and are curved, and when they bump into each other, they capture each other in a vertex. And the curvatures of those vertices are similar to curvature itself. But they might be also just because that's the scale they grow on. So, but here what I showed you is wild type, and wild type is quite straight.
So the 1D model is great, and I've also used 1D models to model vesicles. But I'm wondering, in reality, it's a bilayer, right? It's a bilayer, and there can be chemical asymmetry or other effects that are in the leaflets. I do have a 2B thick model that might be helpful. So the question is, are you aware of any mechanisms that if the 2B thick, basically two layers modeled separately would give you that is not already included in the 1B thick model?
So, I mean, somehow whenever we get our paper back from reviews, we get a question on the model. And then we always have a bit of work repeated with not the 2-bit model, but the Zerno's 3-bit per lipid model, which is this just because we happen to have that coded up, and it's also now in Lambs. And then, so for membrane reshaping, it's still much lower model, so you cannot do the full exploration of the mechanisms and so on. But once you know the mechanism, you can repeat kind of the main thing, and it still looks the same. I think decision stat is probably that mechanism, if that model is any better, because it's still quite phenomenological. And the problem is a bit that we have no experiments also on this, the real scission stat. It's below the resolution. So I don't know which model maybe better would be and more appropriate for that. But also none of the conclusions we have here are based on this last step decision. It's more on the first part. But I should say something, just one thing to make them even more uncomfortable. So do you know, so archaeal membranes, they're not bilayer, at least not those, they're extremophiles, they're monolayer. They look like the lipids. They look like
Oh,
yes. And it's known that extremophiles, both archaea and bacteria, have the higher temperature than even the more of these they have. This fluid phase is higher temperature. So now you may ask questions, how is this being cut and fused, right? Most probably, never been this situation. So you get some kind of local curvature, local bending of a lipid, boiler lipid, and then you kind of recreate effectively bilayer. And then we do know also that they have some small influences of bilayer, but a couple of % only. So it could be that these guys go on the highly curved regions to enable it to reshape and reform. But yes, I think we've all been ignoring this. And it's also unclear how our machinery comes from archaea that have these very different members. So they're also chemically different. They're not phospholipid, they're tetraether. So yeah, there's this called lipid divides. Kind of, we have lipids from bacteria, but machinery from archaea.
Very interesting food for thought. Obviously, you learn more about archaea.
We also,
But one filament is very important in eukaryotic cell division, which again, textbooks are probably being rewritten to work more now, but is septins. So that forms filaments, in addition to cross-linker. And so is it known that archaea, whether your archaea have septins?
question. I don't know. I've never heard, so which might not mean that they don't. I've never heard discussion on septins in archaea. Yeah, and I don't know. So I mean, I know they're there in this bridge scission. They're also related to microtubule scission because they're in this bridge.
Another really interesting thing about septin is it's an apolar filament.
Exactly.
So therefore, wouldn't presumably be, possible to have an active miasm machine.
Yeah, but it's actually still binds GTP, but it doesn't hydrolyze. So
Any more questions? Okay, so I think let's thank people.